Right now, pharmaceutical companies are spending like billions of dollars trying to find the perfect molecular wrench, just to fix a single broken gear in your cells.
Yeah, and it's a completely exhausting process.
It really is. But what if we've been looking at the human body entirely wrong this whole time? What if the secret to curing disease isn't about fixing one tiny broken gear, but rewriting the cell's entire software code?
Well, that is the exact paradigm shift we're seeing right now.
Welcome to today's deep dive, customized just for you. If you've ever wondered why so many promising drugs just fail before they ever reach your pharmacy, well, today we actually have an answer. We are pulling apart a massive, brand new 2025 research article from the journal Science.
And this paper, it is a heavyweight. It details an artificial intelligence framework called DrugReflector.
Right.
And what we are dealing with here is basically a fundamental shift in how we discover medicine.
So the mission for our deep dive today is to explore exactly how this lab-in-the-loop AI completely abandons the old way of doing things. Nice. Instead of looking at just one protein, it uses the entire genetic signature of a cell to predict which drugs will actually work.
Exactly.
And spoiler alert for what's to come here by doing this, it boosted drug discovery success rates in the lab by an astonishing 13 to 17 times.
Yeah, and I want to emphasize that. In the pharma world, a 13 to 17-fold increase, that isn't just an incremental update.
No, not at all.
It is a seismic, paradigm-shattering leap.
But, to truly appreciate what DrugReflector is doing, I think we first need to look at the wreckage of the current system. We mentioned that broken deer approach earlier. Why is that failing us so spectacularly?
Well, simply put, because a human cell is not a simple machine like a clock. It is a highly complex, dynamic ecosystem.
Right, it's alive.
Exactly. For decades, the traditional approach has been what we call single-target drug discovery.
Yeah.
So a researcher finds a specific protein that seems to be misbehaving and causing a disease. So they spend years designing a chemical just to block that one specific protein. But here is the fatal flaw in that logic cells have evolved over billions of years to survive at all costs.
Yeah, they're resilient.
Highly resilient. They have these incredibly robust, redundant mechanisms built into them.
So if you block one pathway, the cell just, reroutes the traffic. It finds a side street.
Precisely. The cell simply uses a compensatory mechanism to keep doing whatever it was doing. And your single-target drug, well, it essentially becomes useless.
Man, that's frustrating.
It is. And to make matters more complicated, there's a biological reality known as polypharmacology.
Polypharmacology. Okay, break that down for us.
Sure. So most chemical compounds are actually quite messy. They don't just hit one target. They interact with multiple proteins and pathways all at the exact same time. The single-target approach tries to pretend this messiness just doesn't exist.
Wait, let me push back on that for a second. Because if a drug hits multiple pathways and creates this massive biological ripple effect, isn't that exactly what causes terrible side effects?
That's the standard thinking, yeah.
Right, so how is that messiness a good thing? If we embrace that, how do we know the drug isn't curing the disease, but ultimately killing the patient?
That is the million-dollar question right there.
Yeah.
And it's exactly why the alternative approach, which is phenotypic screening, is so vital.
Okay, phenotypic screening.
Right. Phenotypic screening basically says, stop looking at the individual proteins. Instead, it looks at the observable traits, the phenotype of the entire cell.
So you're looking at the big picture.
Exactly. You apply the messy drug, and you simply ask, does this cell look healthy now? Is it behaving normally? It accounts for all those ripples and unintended interactions because you are only judging the final net outcome on the whole system.
I always think about this using a car analogy. The old single-target approach, it's like trying to fix a strange rattling noise in your engine by blindly replacing one specific spark plug.
And just crossing your fingers.
Exactly. Crossing your fingers and hoping that was the root issue. But phenotypic screening is like taking the car for a test drive. You don't necessarily care which spark plug or belt is causing the problem initially. You just want to hear how the whole engine sounds and performs under stress.
That's a great way to look at it.
Yeah.
And take that car analogy one step further. You aren't just listening to the engine. You're looking at the exhaust, checking the tire pressure, monitoring the dashboard. You are assessing the entire vehicle.
Right.
And historically, this works. Retrospective studies show that over 65% of our approved drugs were actually discovered through phenotypic observations. not through single-target design.
Wow, 65%. So if you're listening to this and wondering, well, if it works so much better, why isn't every pharma company just doing it all the time?
Yeah, it comes down to logistics and economics. High-resolution phenotypic screening is painfully slow and, honestly, astronomically expensive.
Because of the manual labor?
Yes, the sheer manpower. To do it right, you have to take thousands of live, complex human cells, apply thousands of different chemical compounds to them, and then meticulously observe the physical results under a microscope over a long period of time.
Oh, wow. So doing that for millions of compounds is just impossible.
Completely impossible to scale up. We need a proxy, a shortcut that gives us the whole cell picture without the massive physical cost.
And according to the science paper, that proxy is something called transcriptomics.
Yes. So transcriptomics is essentially taking a high-resolution snapshot of all the messenger RNA, the mRNA inside a cell at a specific moment in time.
Okay, the mRNA.
Right. The mRNA tells us exactly which genes are turned on, which are turned off, and like the volume level of each one. It's reading the cell's entire active software blueprint.
Got it.
So if we can see the transcriptomic signature of a sick cell, and we know the signature of a healthy cell, we just need to find a drug that forces the cell to transition from the sick signature to the healthy one.
Okay, causally speaking, that totally makes sense. Transcriptomics gives us the whole picture of the cell's software, but we still have, billions of possible chemical compounds out there in the wild.
We do.
How on earth do we match a specific chemical to the exact picture we want to create?
Well, that is where the researchers introduced DrugReflector. It's an ensemble of deep learning AI models designed to act as a highly trained translator.
A translator between what and what?
Between the language of chemistry and the language of cellular biology. But, as you know, for an AI to translate anything, it needs a massive dictionary to learn from.
Right, and AI is only as smart as the data you feed it. So what was the training data they used here?
They trained DrugReflector on a massive database called the connectivity map, or CMAP for short.
CMAP, okay.
Scientists have previously taken 9,597 different chemical compounds and physically tested them across 52 different human cell lines.
Wow, that's a lot of data.
It is. They recorded the exact transcriptomic signature, all the gene expression changes caused by every single one of those perturbations. By feeding this into the AI, DrugReflector learned to associate specific molecular structures with the exact cellular ripples they cause.
So the AI basically learns the cause and effect. Like if I drop chemical X into this environment, genes A, B, and C turn up, and genes X, Y, and Z turn down. But wait, how does the AI mathematically understand what a cure actually looks like? I mean, we know what sick looks like, and we know what healthy looks like, but how do you give an algorithm a solid target to aim for?
The researchers solved this by developing a brilliant mathematical metric. They called it the V-score.
The V-score.
Right. Imagine a three-dimensional scatter plot. On one side, you have a cluster of points representing the gene expression of the sick cell. On the other side, you have a cluster representing the healthy cell.
Okay, I'm visualizing that.
The V-score is the mathematical calculation of the statistical difference between those two states.
Yep.
It is a highly specific, multi-dimensional vector.
Oh, I see. It's essentially like a GPS system calculating the route between two coordinates. The V-score is the exact biological delta, the literal route we need to travel.
That is a perfect way to visualize it.
Yeah.
You hand that V-score coordinate to DrugReflector and essentially command and scan your entire understanding of chemistry and find me the compound whose biological ripples will perfectly bridge this exact gap.
That's wild.
And before they ever touched a live cell, they computationally benchmarked this. They ran DrugReflector against a massive data set of 1.2 million cells across 10 different cell lines.
And how did it do?
It consistently and significantly outperformed 4 state-of-the-art alternative algorithms.
Okay, computational benchmarking is one thing. Playing around with math in a database is safe, right? But human biology is incredibly messy.
Very true.
If we take this AI's predictions and put them into an actual wet lab with living, breathing cells, does it just completely fall apart?
The researchers wanted to answer exactly that. So they set up the ultimate proving ground, a blood cell test. Specifically, a process called hematopoiesis, which is the creation of new blood cells in the body.
Okay, hematopoiesis. What kind of blood cells were they trying to create?
They had 2 distinct targets. First, they wanted to find drugs that could create red blood cell progenitors, which are called erythroid cells, to treat things like severe anemia.
Got it. And the second?
They wanted to find drugs that could create platelet-making cells, known as megakaryocytes, to treat blood clotting deficiencies.
And what starting material were they using? Because you can't just conjure blood out of thin air in a lab.
No, you can't.
Yeah.
They used primary CD34 positive hematopoietic stem cells.
Let's pause there for a second, because using primary stem cells, that is a massive detail in this study, isn't it?
Oh, it is arguably the most crucial detail. Primary stem cells are sourced directly from human donors. They are not immortalized cancer cells that have been just growing in a lab for decades.
Right, they're the real deal.
Exactly. And primary cells are incredibly finicky. They are difficult to keep alive, and they are exorbitantly expensive to culture. Most early drug discovery avoids them entirely because it's just too hard.
Wow.
So by using primary stem cells, the researchers were taking on a massive experimental challenge to prove their AI works in the most clinically relevant, true-to-life setting possible.
So we essentially need to find a needle in a haystack, a chemical that will force these delicate, expensive stem cells to mature into either red blood cells or platelet factories. What is the normal industry standard baseline for doing this without an AI?
The standard is literally called brute force screening. You take a massive library of random compounds, use robotic arms to drop them into thousands of tiny wells containing the stem cells, and you just wait to see if anything happens.
Just guessing and checking.
Pretty much. In this study, they ran a brute force screen to establish a baseline. Out of all the random compounds they tested, the hit rate was a dismal 1%.
1%. So you test 100 drugs, you burn through thousands of dollars of primary stem cells, and you get maybe one chemical that does what you want.
Yeah, it's highly inefficient.
Think about the wasted time, the wasted resources. I mean, no wonder clinical trials are so incredibly expensive.
But then they handed the reins over to DrugReflector. They gave the AI the V-score, that transcriptomic gap between the stem cell and the mature blood cell, and asked it to rank a list of compounds it predicted would actually work.
And what happened?
When they physically tested the AI's top picks in the lab, the hit rate for the platelet-making megakaryocytes was roughly 20%.
Whoa.
And for the red blood cell erythroid progenitors, it was 16%.
I really want to make sure you listeners absorb those numbers. Going from a 1% hit rate to a 20% hit rate is a 20-fold leap. Achieving a 13 to 17-fold increase over random chance completely changes the fundamental economics of drug discovery.
It absolutely does.
It means a lab can actually afford to use those highly accurate primary stem cells because they aren't just wasting them on thousands of dead-end chemicals.
It fundamentally de-risks the early stages of discovery. But, here is the catch. If you have a 20% hit rate, that means the AI was still completely wrong 80% of the time.
Right, and that is actually where the story gets even better. Because if I'm a researcher and 20% success rate is, like, cause for popping champagne, but these researchers looked at the 80% failure rate and asked, why?
Exactly, which brings up a critical lesson about biological context. Remember the training data we talked about earlier, the CMAP database?
Yeah, the 9,000 compounds.
Right. Well, it was built almost entirely by testing drugs on immortalized cancer cell lines. It did not contain any data on how drugs interact with primary CD34 positive stem cells.
Ah, so the AI had a massive blind spot. A chemical might cause one reaction in a breast cancer cell and a completely different reaction in a healthy blood stem cell. The AI was making highly educated guesses, but it was essentially speaking a foreign language. So how did they fix that without throwing the whole model out?
They introduced a closed feedback loop, a process they called active reinforcement learning. This is the core of that lab-in-the-loop concept.
Okay, how does that work?
Instead of just throwing away the 80% of drugs that failed, they took the hits and the non-hits from that first physical experiment and ran them through single-cell RNA sequencing.
This mechanism reminds me exactly of how a high-end music streaming algorithm works. Like when an app suggests a song and you immediately hit skip, a good algorithm doesn't just register a failure and move on. It analyzes the metadata of the failure. It asks, why did you skip it? Was the tempo too fast? Was it a jazz track when you usually listen to rock? Were there too many acoustic guitars?
Right.
It actually uses the failures to refine your specific, highly nuanced case profile.
That analogy hits the nail right on the head. The researchers took the paired data, meaning the actual physical results of the cells, combined with the deep transcriptomic RNA data of both the successes and the failures, and fed it all back into DrugReflector.
Wow.
They essentially told the AI, here is where your prediction matched reality in these specific stem cells, and here's exactly how your prediction deviated from reality.
They forced the AI to recalculate the GPS route based on real-world traffic conditions. They refined that V-score signature. So what happened when they ran a second batch of compounds using this newly educated, actively learned AI?
The hit rate doubled again.
No way!
Yes, but even more impressively, the median rank of the true hits, meaning how high up the AI placed the winning drugs on its priority list, jumped from 463 to 138.
That is incredible. It proves that the model isn't static. It can learn, adapt, and optimize itself for entirely new biological contexts with just a single round of physical feedback.
And because transcriptomics gives us such a detailed view of the cell's inner workings, the AI didn't just hand the researchers a better list of drugs. It actually forced them to uncover entirely new human biology.
Okay, let's definitely dig into that. How does an AI accidentally teach human experts about our own biology?
Well, the AI is effectively showing its mathematical work. When the researchers looked closely at the gene signatures of the drugs that successfully forced the stem cells to become platelets, the megakaryocytes, they realized those successful drugs fell into two distinct mechanism of action classes.
Meaning the drugs were doing two completely different things inside the cell to achieve the exact same result. What was the first group doing?
The first group were classified as strong inducers. When they investigated the molecular targets of these specific drugs, they found that they were blocking multiple tyrosine kinases at the exact same time.
Tyrosine kinase.
Yeah, specifically proteins known as KDR and FLT3.
For those of us who aren't cellular biologists, what exactly is a tyrosine kinase and why does blocking them matter?
Think of a tyrosine kinase as a cellular antenna. It sits on the surface of the cell, receives chemical signals from the outside, and relays that message deep into the cell's interior to tell it how to behave.
Makes sense.
Now, normally, a human drug designer might try to design a hyper-precise drug that hits just one of those antennas perfectly to avoid side effects.
Going back to that single target approach we talked about.
Exactly. But the researchers decided to test if that would actually work. They used CRISPR gene editing technology to physically knock out just one of those kinases. And the result was that the stem cell didn't fully transform. The single target approach completely failed.
Wow. So the AI proved that the messy polypharmacology, the simultaneous blocking of several different antennas at once by these specific drugs, was actually the biological key to driving the cell's fate. It wasn't a side effect, it was the mechanism.
Exactly. It was a feature, not a bug. But the second group of successful drugs, the moderate inducers, provided an even bigger shock. When they looked at what these drugs were targeting, they all pointed to a single protein called HMGCR.
HMGCR? I have no idea what that is. Why is that shocking?
Well, HMGCR is a master regulator of cholesterol biosynthesis. It is the exact same protein that common statin drugs target to lower your cholesterol levels.
Wait, lowering cholesterol helps create blood platelets? That sounds completely unrelated.
That was the reaction of the biological community as well. Until this AI pointed it out, lipid metabolism and cholesterol pathways were not widely appreciated by human biologists as being a primary driver of early megakaryocyte commitment.
That is wild.
Right. The AI, simply by reading the raw math of the gene expression, found a completely unexpected biological backdoor to trigger platelet production. It found a hidden lever that human experts hadn't prioritized because of our own historical biases about how blood is supposed to form.
That is mind-blowing. It's literally reading the code better than the people who discovered the code. But obviously, a framework like this needs to do more than just make blood cells if it's going to truly revolutionize medicine. Did they test DrugReflector on more complex diseases like, say, cancer?
They absolutely did. They pointed DrugReflector at two incredibly aggressive forms of cancer: B-cell acute lymphoblastic leukemia, known as BAL, and triple-negative breast cancer.
Okay.
In both cases, they fed the AI the transcriptomic signature of the cancerous state and the signature of the healthy state and basically said, find the chemical bridge.
And what did it prioritize for those? Did it hallucinate random chemicals or did it find real oncology drugs?
For the BL leukemia, the AI's top recommendations included a drug called ponatinib. And what's crucial to understand here is why it chose ponatinib.
Why did
the AI identified that the leukemia cells had a massive dysregulation in a specific kinase signaling pathway. Ponatinib is a known potent inhibitor of that exact pathway. In fact, it's an actual clinical standard of care drug currently prescribed by doctors to treat that specific leukemia.
So it organically reconstructed the standard of care from scratch just by doing the math on the RNA.
Exactly.
What about the breast cancer?
Triple-negative breast cancer is notoriously difficult to treat because it lacks the usual hormone receptors that drugs target. But when DrugReflector read the transcriptomic signature, it saw massive dysregulation in the cell's division pathways. The cells were trapped in a hyperactive mitotic state, constantly dividing.
Right, which is what cancer does.
So the AI strongly prioritized a class of drugs called cell cycle inhibitors. The specific V-score of those inhibitors perfectly counteracted that hyperactive division state. It read the specific vulnerabilities in the RNA and prescribed the exact mechanism needed to halt the cancer.
Man, it really validates that the framework isn't just a party trick for blood cells. It can translate across vastly different, highly complex disease contexts.
It proves that if you can accurately map the transcriptomic gap between sick and healthy, DrugReflector can find the chemical to cross it.
If you are listening to this, let's take a quick step back and recap the incredible journey we just took. For decades, modern drug discovery has been stuck in a highly reductionist mindset.
Yeah, very narrow.
We thought of the human body as a simple clockwork machine, spending billions to find a single chemical key to fit a single protein lock. But cells are incredibly complex, redundant, and messy ecosystems.
And to cure diseases, we needed a way to measure the net effect of a drug on the entire biological system. But doing that physically in a wet lab was just too slow and way too expensive.
So the scientific community developed a proxy transcriptomics, reading the messenger RNA to see exactly what the entire cell is doing at any given moment. And then these researchers built DrugReflector, an AI trained on thousands of chemical reactions, capable of mathematically mapping the exact distance between a sick cell and a healthy cell using that multidimensional vector, the V-score.
Which, amazingly, took us from a dismal 1% success rate to a 20% success rate in some of the hardest to culture primary stem cells in the world.
And crucially, when it failed, it didn't just give up. Through active reinforcement learning, that Spotify-style lab-in-the-loop feedback, it learned exactly why it failed, doubling its accuracy again on the fly. And along the way, it taught human experts completely new things about how cholesterol affects blood clotting.
It is a fundamental shift in perspective. We are finally moving away from trying to outsmart the cell's individual proteins and moving towards speaking the cell's holistic language.
And that leaves us with a really provocative final thought to mull over today. DrugReflector successfully discovered that manipulating cholesterol pathways could cure platelet deficiencies, which is a deep, complex connection that human experts hadn't made. As these AI models become even better at reading the multidimensional language of cell states, it forces us to ask a massive question about the future of medicine. Are we rapidly approaching an era where we no longer invent drugs in the traditional trial and error sense? Are we moving toward a future where a doctor simply defines the cellular code they want to rewrite, and we just ask an AI to compile the drug for us?
Given the massive leaps in hit rates we're seeing here, it certainly looks like that's where the science is heading. We are transitioning from finding the right wrench to writing the right biological code.
And that is a fascinating place to leave it for today.